Today, I will introduce the fascinating world of multisensors and their importance in healthcare. The goal of multisensors is to integrate several sensors to collect relevant data on different health parameters. This approach is multiscale and focuses on understanding the functioning of organs at microscopic level above the centimeter, to the microscopic cellular level, and even at the nanoscopic scale to understand the mechanism of operation. Health tracker sensors are mostly physical and were developed in the 70s. For example, to detect an infection, we use temperature measurements, while pressure and electrocardiograms are used to measure blood pressure and heart activity. Optical measurements are used to monitor the oxygen levels, while electrochemical sensors measure glucose levels. These sensors were developed in a sensitive way and even at low cost more than 30 years ago. However, the challenge with these sensors is the accuracy and the reliability, as they are not specific to any particular disease and interferences with other parameters can occur. This is where multisensors come into play. They improve reliability by measuring several parameters at the same time, and the correlation between these parameters has been studied since the 2010s. By measuring the pressure and oxygen levels, for example, the reliability can be increased by 90% for certain pathologies. Wearable sensors connected to Internet of Things or our mobile phones allow low-cost monitoring of several sensors for short-term measurements. However, for long-term measurements and to gain sensitivity, sensors can be implanted as close as possible to the target organ, allowing real-time measurements. Despite the high cost of these sensors, which can be prohibitive, efforts have been made in the last decade to develop personalized diagnostics based on artificial intelligence, making use of the vast amount of information collected by multisensors. Let's move on to the second scale, which is cellular. This involves counting cells in the blood, including red and white blood cells and platelets. The concentration of red cells is usually over 1 billion cells per liter, with the size considered being around 10 microns. The first technique developed to count cells is electrical, dating back to 1950. This technique measures the electrical resistance of a small hole, allowing us to detect single cells with a reading speed of around 1,000 cells per second. The second technique, which is optical, dates back to 1968 and is called flow cytometry. This technique allows us to detect single cells with the same sensitivity as the electrical technique, but with the added benefit of using fluorescence markers to identify subgroups within a complex population. This is known as multiplexing. Both techniques are more expensive and can detect a single abnormal cells among 10,000 cells, making them useful for detecting pathologies such as cancer. However, the current challenge is to detect an abnormal cells among 1 million cells for early detection purposes. This is where the nanometric scale comes into play. The field of nanotechnology has revolutionized various aspects of science and technology, including the development of nanosensors for DNA sequencing. With a multiplexing of 900,000 different DNA probes immobilized on the surface, nanosensors can sequence the entire human genome, which used to take decades to accomplish in the Human Genome Project program. This advancement has drastically reduced the time needed for DNA sequencing to just a few days, with a reliability rate of less than 0.01% and the cost of only €1,000 per chip. Another remarkable example of nanosensors application is in the detection of COVID-19 virus. The PCR test, a technology developed in the 80s, is the most reliable test for detecting the RNA present in the center of the virus. This test replicates strands exponentially, enabling the detection of even very low quantities of virus, just a few copies per milliliter. All this is accomplished at a cost of only 50 euros per test, with a response time of just 1 hour. Even faster and cheaper diagnostic options are now available. This enables the self-testing and decentralized diagnosis. The principle is to detect the foreigner proteins in the body that are fixed on the surface of the virus. Those immunoassays, which have been around since the 50s, are well proven in pregnancy tests and have been developed to detect antigen with excellent specificity rate. This is achieved through the use of antibodies that sandwich the antigen. Then a transducer is used to detect the sandwich. The sandwich is formed during the fluid migration over functionalized areas. This technology provides qualitative results within minutes with a specificity rate of 97%. You might say, we found the perfect sensors, ultra-sensitive, inexpensive and specific in the field of genomics. However, the medical research sector is currently facing a reproducibility crisis. A survey done by the prestigious journal Nature shows that 47 publications among 53 were not reproducible on the subject of the cancer. This is partly due to the correlation effects among hundreds of variables which can transform non-significant results into positive ones. One of the most widely publicized examples of this crisis was the scandal in Silicon Valley. The company Theranos, valued at $10 billion, claimed first to have developed revolutionary technologies capable of measuring cholesterol, glucose, and hormones levels in a single drop of blood. However, the results were completely distorted by cross-reactivity with biotin and vitamin D, as well as lies about the protocol used, with the only purpose to raise fundings. These scandals highlighted the importance of accuracy and reliability in blood testing technologies. ROC curves are used to evaluate the accuracy of tests based on selectivity and sensitivity of signals. The current sensors used to prevent heart attack are not effective enough for routine use in laboratories. And even the results obtained in the laboratories are not exceptional. Only 30% of patients can be detected. But point-of-care sensors are even less efficient. There are many reasons for this lack of selectivity. From a chemical perspective, the functionalization of sensors is an old idea dating back to Langmuir or more or less the 50s. The surfaces are never perfect, and the heterogeneity of the surfaces combined with the complexity of the liquid medium leads to chemical interferences. Detecting the proportion of a mixture of 4 molecules still remains a challenge due to the competition between affinities. From a biological perspective, that antibodies are flexible molecules that can adapt to the wide variability of antigens, creating fluctuations that depend on the environment, pH, temperature, ionic strength. To address these issues, several approaches have been developed. Disease can be represented as observables, and at the biochemical level, we need a multiplex approach, namely to have multiple fingerprints of these states. We have to target a panel of proteins that serves as biomarker. The second idea is the multisensors, which are miniaturized and can be optical, electrochemical, mechanical, or magnetic. While all sensors have equivalent cost and quite the same sensitivities, the limit of detection has reached a target of the order of picomolar. However, Raman spectroscopy enhanced by plasmonics is one of the most sensitive techniques capable of detecting individual proteins with unique spectroscopic fingerprints. The current trend is to improve the reliability of medical sensors through statistical analysis and correlation between the signals of each sensor that prove different physical or chemical properties. By doing so, we can increase the level of confidence in these sensors. In conclusion, we have seen the trend in multiscale, multiplexing, multisensing. Nanotechnology is still the field that holds enormous potential for early stage detection of disease. As we move forward, we must continue to prioritize accuracy, reliability, and reproducibility to ensure that we are delivering the best possible care to patients. Thank you for your attention.